Hello friends, so in the last lecture we discussed the electron transport or ETS, electron transfer system. Okay, now in the electron transfer system the electrons are emitted by PS2 system. Okay, and then the electrons accept by electron acceptors which are present in the lamellae of thylakoid or membrane of thylakoid. Then they transport through a different electron acceptors or the molecules who receive these electron and release the electrons. Okay, then the ATP and NADPH2 is synthesized, and simultaneously the water molecule is also splitting. Okay, then now this is the common photophosphorylation reaction, rather this is the common electron transport system. Now due to the sun rays or due to the sunlight or due to this photon which is present in sunlight, there are two different wavelengths are absorbed by two different photosystem. Okay, the first one is, हम पहली बात करते हैं, the first one is PS1 which is stimulated by 700 nanometer wavelength. So if sunlight of or the wavelength of light is 700 nanometer which is absorbed by photosystem 1. Okay, so photosystem 1 absorb this wavelength and they emit the electron. Okay, 4 electron rather, they emit the electron. This electron are accepted by electron receivers or electron acceptors. Okay, electron acceptor. Okay, they receive the electron. Okay, and then वो खुद का redox करते हैं. They receive the electron and reduction होता है, and they release the electron and they give this electron to a different ETS, electron transfer system. Now what is the part of this ETS, or which are the molecules of this ETS? As I earlier said, there are the quinone, plastoquinone, ferredoxine, plastocyanine, cytochrome A, cytochrome B, cytochrome C in the different manners, okay, or in the different arrangement. So there are the different cytochromes are there, and then the 4 electron is released after ETS. Now what is the next thing? So if you talk about the PS2 system is present in the thylakoid as well as in the interlamellae or interthylakoid lamellae, okay, or intergranum lamellae you can say. So उन दोनों के अंदर PS2 है, but PS1 is only present in thylakoid membrane only or thylakoid only. So there is no NADP reductase enzyme present in the external lamellae, और बाहर जो lamellae है वहाँ पे inner side the NADP reductase enzyme and PS2 is not there. So these 4 electrons are back into the PS1 system. वो PS1 system में वापस चले जाएंगे because there is no NADP reductase enzyme. So जो 4 electrons which is arise from PS1 system and they back to the PS1 from after the traveling of electron acceptors and ETS, and after the synthesis of 1 ATP, so they return to the PS1 system. Means वो PS1 system से निकले और पूरा चक्कर लगा के they are cyclic phosphorylation, that's why वो पूरा चक्कर लगा के वो PS1 में वापस आते हैं, that's why it's called cyclic photophosphorylation. Now during the ETS they transport the different they transport the electron through a different molecules. During this transportation of electron, the phosphorylation process occurs. Now this phosphorylation process, they done this phosphorylation process वो क्या करते हैं, ATP में ADP में एक इनऑर्गेनिक फॉस्फेट को ऐड करके दिस फॉस्फोराइलेशन प्रोसेस अकर्स एंड दे फॉर्म द ATP, दे प्रोड्यूस द ATP rather इन द एनर्जी फॉर्म। एनर्जी फॉर्म के अंदर ATP प्रोड्यूस करते हैं, ओके सॉरी ATP प्रोड्यूस करते हैं इन एनर्जी फॉर्म, सो दे प्रोड्यूसेस द ATP, ओके। सो ये जो पूरा साइकिल है उसको बोलते हैं साइक्लिक, मींस इलेक्ट्रॉन जो पूरा साइकिल कवर करता है, दैट्स व्हाई इट्स कॉल्ड साइक्लिक। एंड फोटो, because of this photon and due to the transportation of electron, phosphorylation process is there, that's why it's called photophosphorylation, okay, cyclic photophosphorylation. Now second method means the photosystem 2, as you know, and we saw the early lectures. If you see the early lectures, PS2 is already there, so it is simulated by 680 nanometer wavelength, okay. They emit the 4 electrons, okay, they emit the 4 electrons, and there is the electron acceptor जो इलेक्ट्रॉन रिसीव करता है, then they goes to the ETS, and after the ETS this 4 electron is absorbed by PS1, or rather they comes into the PS1, okay, और PS1 से वो रिलीज होके फिर से ऊपर जाते हैं इलेक्ट्रॉन एक्सेप्टर्स के पास, एंड देन वहां पे NADP रिडक्टेज एंजाइम है जो इंटरलैमेली के अंदर है, नहीं है अगर, और NADP कन्वर्टेड इनटू NADPH2, ये H2 कहां से आया और ये इलेक्ट्रॉन कहां से आया वो भी मैंने आपको बता दिया था। इलेक्ट्रॉन इस चैनल से आए हैं, H+ है वो वाटर स्प्लिटिंग से आए हैं, एंड O2 रिलीज हुआ, ओके, एंड दे क्रिएट अ Z स्कीम, जिसके बारे में हमने बात की थी प्रीवियस लेक्चर। Z स्कीम में भी नाम रह गया था, तो वो ड्यू टू दिस channel or due to this pathway uphill and downhill transportation, it's called Z scheme. So you can easily observe Z scheme in photosystem 2, but there is no Z scheme in photosystem 1. तो photosystem 1 के अंदर Z scheme नहीं है, photosystem 2 में Z scheme है। Now the differences between PS1 and PS2. Second thing, here the ATP is also there, और ATP is produced, यहाँ पे भी ATP बनता है, यहाँ पे भी ATP बनता है, it is common thing, but यहाँ पे बनता है NADPH2, यहाँ पे there is no formation of NADPH2 because the absence of NADP reductase in the PS1 के अंदर आपको NADP reductase नहीं मिलेगा, that's why there is no production of NADPH2, okay. ATP is there, ATP दोनों के अंदर मिलता है। Now the differences is water splitting occurs in the PS2 or non-cyclic photophosphorylation, or there is no water splitting in PS1 क्योंकि उसके इलेक्ट्रॉन्स वापस उसमें आ जाते हैं, that's why there is no need to give the electron to PS1, okay. So water splitting नहीं होता PS1 में, या cyclic photophosphorylation में water splitting नहीं होता, non-cyclic photophosphorylation में water splitting होता है, ओके। इसमें Z स्कीम बनती है, इसमें Z स्कीम नहीं बनती, इसमें NADPH2 बनता है, इसमें NADPH2 नहीं बनता। सो दीज आर दी differences of cyclic and non-cyclic photophosphorylation, okay. Now due to this cyclic and non-cyclic photophosphorylation, NADPH2 and ATP बनता है, okay. So ATP formation होता है in the thylakoid membrane, okay. Or now due to this cyclic and non-cyclic photophosphorylation, there is the differences occurs, or there is the formation of ATP and NADPH2 तो है जो हम यूज करेंगे in the further to biosynthesis phase और other dark phase के अंदर, कि जो हमको फूड फॉर्मेशन या ग्लूकोज फॉर्मेशन में हेल्प करता है NADPH2 and ATP, but there is the another concentration differences occurs between the in the chloroplast. What is that? So उसके बारे में बात करते हैं कि as you know कि there is the, if it is the thylakoid, for an example this is thylakoid, ओके, सो थाइलेकॉइड के अंदर होता है, इसके मेम्ब्रेन पर ये जो ETS है वो दोनों है. सो PS1 and PS2 both is occurs in the thylakoid lamellae, PS1 and PS2. Now there is the different, due to this photophosphorylation and due to this water splitting and so many methods, यहाँ पे in the thylakoid lumen of this thylakoid, the H plus or hydrogens are accumulated. Means concentration of H plus ion become increases in the thylakoid lumen. Or parallelly, the stroma, this is the stroma. So stroma के अंदर there is the low concentration of H plus ion, and here is the high concentration of H plus ion. Now, how it occurs and why this low concentration and high concentration is there, and it creates the proton gradient, or they produces the proton gradient or different concentration of proton. That's why it's called chemiosmotic hypothesis or chemiosmotic method or chemiosmotic rules. Okay, कि यहाँ पे प्रोटॉन के concentration different है due to this different concentration gradient. The concentration gradient is there due to this concentration differences, and there is the membrane of thylakoid membrane. So उसकी वजह से एक membrane है और दो different concentration है, so वो osmosis produce करता है. And here the proton gradient is there, that's why it's called chemiosmosis. Okay, now what is the significance of chemiosmosis? As you know कि due to the photophosphorylation you generate the ATP, and now this chemiosmotic method or chemiosmotic जो process है, इसकी वजह से भी the ATP energy production is there, and transportation of proton ion, or you can say H plus is also called proton. So transportation of this proton ion is also helpful in the formation of ATP. So इसके बारे में डिटेल हम स्टडी करेंगे इन द नेक्स्ट लेक्चर. सो आज यहाँ तक रखता हूँ. साइक्लिक एंड नॉन साइक्लिक फोटोफॉस्फोराइलेशन एंड बेसिक इंट्रोडक्शन ऑफ कॉन्सेंट्रेशन ग्रेडियंट. ओके, प्रोटॉन कॉन्सेंट्रेशन ग्रेडियंट एंड दे क्रिएट द केमिओस्मोसिस. ओके, सो यहाँ तक रखता हूँ. नेक्स्ट लेक्चर में देखते हैं व्हाट इज केमिओस्मोसिस. ओके, सो आज यहाँ तक रखता हूँ. ओके, बाय.